Buenas tardes.
La cámara está.
Our speaker today is Ignacio Obeso. Ignacio is a behavioral neuroscientist. He works in the Neuroscience Center Cajal. The name it has changed. Neuroscience Center Cajal is the current name. And he's the director of the group Cognitive Control and Habit. Why habit and not behavior?
We study a lot habit formation.
Yeah. And well, he's going to tell us about his research. And we thank you very much for coming again.
Yes.
The only excitement we'll have today is your talk, nothing else. Well.
Thank you. Thank you, Pascual, for the invitation and for being here. And it's a pleasure to be back with light. And last time it was another type of excitement. But so, yeah, as Pascual was saying, we are interested in how the brain deals with controlling of behavior and how these behaviors turn automatic and how can we do them without much effort. So that's why habitual behavior is part of the talk. And we've been exploring this behavior in different methodological scenarios. One is experimentally with lab-developed paradigms and also real-life forms of habits. So I will explain you a bit these two sides of the same coin, we think, but to investigate more into the pathophysiology of Parkinson's disease. So in the lab, what we do is different research directions with behavior, neuroimaging, neuromodulation, and clinical as a research pathway. So cognitive control and how they interact with other processes, as well as habit formation, which is the topic of today, and some other. But yeah, the topic is how we can measure in humans habits from outside or inside the lab. Basically, much of our daily behavior, if you are learning something from scratch, you are starting to learn in what we say the habit cycle, and you have an initially goal-directed behavior, which you start to associate your actions with an outcome. So actions that are initially very explorative, let's check here and see what happens. Let's check the plug, see what happens, and you get an electric shock, you won't return there. So your outcome can be positive or negative, and then you repeat it back and forth. And this can then turn into a habit predominant behavior where the stimuli itself, the plug, when I see the plug, I already know what to do. And the trigger, it's my trigger. The trigger is what drives my action. I don't have to think it anymore. It's very highly connected. And the outcome is no longer there. You don't really think what's going to happen with the learned stimuli. And then your action is what you do repeatedly do. If it's healthy, better for you. If it's not healthy, worse for you. So you have a good habit or the bad habit. This is in the popular folk speaking. We get a lot of this, but we investigate that in a more experimental way. So these are mainly guided by two systems, cognitive. One is very goal-oriented, which is driven by the outcome. It's slow, requires effort, and it's flexible. You can adapt it easily. And the other one is the habitual system, which is stimulus-driven, fast. You don't need much effort to do it, but it's quite difficult to revert. It's very rigid in a way. So here we have two situations where we see a very clumsy guy in ice that is mainly in goal-directed system, a more trained person with even can do two things at a time, holding coffee and so on. And there are many different examples where we are in these two situations of goal-directed and habitual system in the daily life. And the brain operates these systems by starting from ventral striatum and prefrontal areas for more goal-directed systems and control. And this transits towards posterior cortical areas and dorsal striatum when you are in this situation, in more habitual performance. If somebody wants to interrupt me, just raise your hand because that will be clear for me where we are all. So basically, two dedicated systems with a transition from anterior to posterior in the cortex and also in the striatum ventral to dorsal. And why are habits so important, at least to our species, is as mostly say, if an act became no easier after being done several times, if the careful direction of consciousness were necessary to its accomplishment on each occasion, it is evident that the whole activity of a lifetime might be confined to one or two deeds. So a man can be occupied all day in dressing or undressing himself if we didn't have this habitual system. So it's a very key and elementary system for our daily behavior. When we were here the last time, this was a very good example for me. I was here when most of you were here as well, with no light. Spain became dark, and we had to look for new routes to get back home, right? So we were all under non-habitual manner. We had to look for other ways of going back home, and that was quite hard to most of us. It was hard to find our way back home. Some share the car with a stranger, and you put into the hands of a stranger, and we successfully managed to adapt our habits in the spatial navigation. So it's a tricky system to play with. If we don't have it, it's difficult to behave. Of course, here at Fundación CIEN and myself, we are interested when these type of behaviors fail and what happens in a situation like the apagón. We don't have anymore our traditional or familiar cues. We have to find for new ones. So in psychology terms, a stimulus-response association drives you to a starting of an action. And basically, you have a stimulus one associated with a response. So let's say a light key, you see the light key, you just go and turn on the light. So this is a stimulus of the light, response to turn on the light. A very kind of simplistic example, but what happens in some addictions is that we have a stimulus one enhanced. Takes a lot of importance, like in addictions or CD, where you have excessive responses. And the other way around would be where stimulus has a reduced association with a response, like in Parkinson's that we are investigated. So depending on some pathologies, you would have different outcomes of these associations. And this can also happen for a stimulus stop behaviors. So a stimulus associated to inhibition, like a red traffic light is the simplest that we all know. We would have, again, stimulus 2 that is associated with a stopping behavior, and this can be increased in depression or apathy. These are all theoretical explanations of how this can be understood in disease, or a reduced link between both stimulus and response, where you get impulsivity or addictions that don't have this access to controlling behavior. So this pattern of how come habits fail is really motivating my thinking and my research activities. And basically, real life examples is when we overconsume caffeine, we are excessively approaching our response to a well-learned stimuli, or when somebody like Cantona loses control over a very also familiar situation like a football match. In the brain, there has been a number of studies looking at how the brain deals with habits, and one key area is the posterior putamen striatum as well. This comes from a lot of rodent studies showing dorsolateral striatum in the human is posterior putamen, also dorsolateral, but more posterior. And this is mainly activated when we express a habit, when I would turn on the light, for example, when it's very consolidated behavior. And it gets connected with premotor cortex, this area around here. The more the posterior putamen connects with the premotor cortex, the better, or the more enhanced, the more consolidated habit we express. This has all been done in humans with a certain experimental paradigms that are not always replicated in the literature. So it's a kind of a field crisis that we are seeing very initial and important studies showing these brain patterns, but are not being replicated, in part because of the paradigms come from animal studies. So the field is moving towards new type of measures. But what we know is that the nigrostriatal system is really or highly dependent on what habits are needed to be expressed, habit formation. Lesions in rats impair how they acquire habits. And also in patients with Parkinson's disease that have dopaminergic deficits in the nigrostriatal system, so how this posterior putamen is not active when they are performing a well-learned task, and they compensate and activate more anterior sections of the striatum. In contrast, the hyperactivity of the posterior putamen has been shown in addictions such as alcohol abuse. So they would be showing higher or increased posterior putamen when dealing with habitual or automatic routines. So we have two types of two very different brain conditions that show different engagement of the putamen during automatic behaviors. So what we can answer or try to move forward the field is how using these type of habits, how overusing them can put humans into a higher risk of brain disorders. Is this a possible risk where we are... ¿Se fue la pantalla? Algo tiene que pasar siempre. Sí. Solucionable, ¿no? ¿Te parece que se ha dormido el proyector? Sí. ¿Te gustó eso? Una pausa no viene bien. Tal cual, ahora ya no sé dónde está. No, 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 no. Es porque estudiamos eso bastante, sí. Sí, sí, tal cual, sí. Sí, sí. Ese es el punto, no sabemos cuánto, pero sí. Un psicólogo médico tipo William James, de la misma época, sí. Sí, pues no lo había pensado, pero claro. Sí, digo yo. No creo que haya muchos más. Sí, por aquí estaba, sí. Vale, gracias. Okay, so I was about to tell about a few of our research questions and based on this topic. So if we overuse the habitual system together with other biological events, we over-recruit this nigrostriatal dopaminergic system, would that put these neurons at risk to degenerate, to die, and therefore arrive at losing some of the habitual functions in some neurodegenerations? So is this a expression of certain disease onsets like Parkinson's or addictions or others? And would these neurons that we know in the nigrostriatal pathway, are they differentially engaged across some disorders of habits, of what we call toxic habits, like, let's say, OCD, that they have very ruminating or repetitive thoughts that can become very toxic? So all these are, of course, very general questions, but we focus on half of them mostly, and specifically with patients with Parkinson's disease that are well characterized for this degeneration of the nigrostriatal system. We are investigating how the deficit of dopamine, which is unilateral, as you can see in this PET imaging, how can that alter differentially habit expression and formation, acquiring a habit and how and expressing it in the real life and in lab developed tasks? So Parkinson's disease, as you all know, is mainly a motor disorder characterized by all these domains of movement, rigidity, tremor, walking, bradykinesia, and other cognitive and autonomic dysfunctions, mainly linked to dopaminergic cell loss and this striatal system, implying motor, cognitive, and limbic circuits of the brain. And all these large network changes after the disease starts also affects other automatic components that have been described since the beginning of the definition of Parkinson's disease, like certain movement patterns that are automatic, like arm swinging or facial expressions, but also automatic cognitive, like flexibility, slowness of thinking, dependency on external cues, and so on. So it sets a very well human model to test how dopamine and habits can be mediating the expression of the disease. So in a opinion letter, we speculated that this transition from a goal-directed system, which is more prefrontal and anterior in the striatum, would be impaired in Parkinson's disease. So if they learn new associations, the transition towards the storage system of the brain, like the habit circuit, would be lost. So they would have less neurons active to perform a habitual task. This has been done in the past with a focus of this topic, and in Parkinson's disease patients doing a very experimental task where they had to learn if a fruit belonged to a right or a left response, and then afterwards they learned these simple associations and they had to reverse the responses, and they could learn pretty well the associations. These are on and off patients compared to controls, and the speed was also faster. This is an older study. They could learn different types of associations. There were 3 categories, but it's not important to go into detail. And a more recent study found that the patients were impaired in both the habit formation when they are acquiring the associations and when they have to reverse them, which is another test. They were also impaired. So they are impaired in anything they do. So quite contradictory. So as psychology is like this, we have always these sort of problems. We tried to answer a bit more specific questions, which is if dopamine and the habit system is down in Parkinson's disease, because we see these automatic movements impairment and cognition as well, they should be impaired as well in when they create and express habits. What if we do experimental habits but longer? So training them for longer, that which they didn't do in this. All the literature they just test patients one day, and they call it habit, which is quite against what a habit can be. So train them for longer and see what happens. So we pick a published paper and task which made patients learn new associations for a sequence of days, and they had to just learn which of these 4 items were associated with these 2 fingers and these 4 left hand movements. By trial and error, they were learning these category associations during 4 days at their home, and then they had some habit tests to check the strength of this learning. And here's where there is a video. Yeah, it's working. You can see that the task, if you get error, it's a red. If it's correct, you get a green. So you won't move until you get the right answer. So that's a way to check that they are learning okay. And I think this was me doing it, and I was not good at the beginning, so, or Parkinsonian maybe, but then at the end they get quite good at it. And so we trained them for 4 days online in their house, and here is not very good, but so in blue you see the patients, and in red the controls. The first day, second, third, and fourth, and they are becoming very stable and no differences in acquiring the learning. So they are unimpaired in acquiring a new association as well, and the accuracy quite stable. So this was during these 4 first days. Then we moved to see how this learning impairs when they have to reverse the learning, when they have to do an opposite thing. So what we call a reversal test, where 2 of these stimuli will change. So now this correct stimuli is associated with a right hand response, and the opposite for this one, now you have to do a left hand response. So just to switch. And then we can get what we call a habitual error, which is to still press right for this one, or still press left in this one. And here, again, we don't see differences, so patients can really still learn to reverse, so they didn't get much of a conflict when reversing previously acquired associations. We see quite similar trials to criterion, accuracy, speed. But what we think that this could be related because they are putting a lot of attention into the tests, so the habit would be similar. What if we just overload the goal-directed system? What if we put them in a dual task so that the expression of the habit is kind of busy and we get the real thing? So here's where we get a bit of a more tricky task. And there will be a video after my explanation, but it's quite what we call freaky test. It's a very dedicated condition, and we put a dual task, which is a counting of tones. So there were 4 tones at the same pace, and basically they had to respond at the 4th tone with the manual response, but according to what they learned. So they could respond in exactly at the time of the 4th tone. That's great, but they could respond too early, so at the 3rd tone, or they could respond too late. They had to basically wait until the 4th tone to do whatever left or right hand was learned. So in this way, we kind of keep busy the attentional system with a dual tone, while we get the behavioral expression a bit more intact in a way. And this is the task. It was quite complex for patients, but they could end up doing it. El sonido, the sound is not there. There's supposed to be a sound like pa, 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 and that's when they have to respond. No hay sonido, no? For every trial, yeah. So it will be pa, 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 and then you have to respond, yeah. But the visual cue could come very early or very close to the 4th tone, that is. So, okay, so they could do it, and what we get is the expression of errors to see how many habitual errors they perform. And then overall, we see that patients can do similar, consistent errors would be just errors to what they previously did. Remap errors are errors looking for a new response, and these are the habitual errors, those that they shouldn't repeat, the errors that were reversed. And when we look at when they had sufficient time in the long time window, the errors are low because they had enough time to produce the response. when they have medium time or short time, we get more errors. And what we see more interesting is that patients in this short time show less errors, less habitual errors than controls. So meaning that they are less in conflict where they have to reverse the previous association. So the habits are inducing them less conflict. They are able to reverse and to produce a newer response. So that's kind of the idea of this task, is that if we put some dual task or we load the attentional system, then patients do less errors because they have less established, less strong habitual system in a way. Is that clear? Yep. Okay, more or less. Yeah. This has been also reported in previous works where they saw like habit-driven errors were reduced in Parkinson's disease. It's not a big effect, as you see here. This is when they were typing in the keyboard texts, they were asked to change some of the words, and they were producing as well less habitual errors. So it's kind of something that it has been there some time. And since this was typing in a typeboard, see.
How does mental flexibility relate to habits? Because can be interpreted as type of habit, right? Mental rigidity.
Yeah. Yeah, I mean, we see it in that way, rigidity of flexibility. So it can be part of this. It can be influencing here. So.
So what that mean then, that in general, like following these findings, patients with Parkinson's disease maybe less stubborn, to say it like that, because they're but they're less mentally rigid.
In a way, we know that patients are more rigid. They tolerate less changes or novelty.
But if we interpret that in a way of habits, because they have less problems, they have less well-formed habits, right?
Yeah.
So they have less problems changing their habits.
Yes.
So this same.
It's in contrast with that idea that they are more rigid, because if we at least for these type of habits that are trained in 4 days, of course, it's a experimental habit. We don't see that rigidity. They are able to change it because we think they were not sufficiently stored or sufficiently associated or sufficiently memorized, if you want.
The interpretation is just that they would need more time to produce an association as strong as the.
Possibly, yeah, as strong as controls, for example, because they have a weaker dopaminergic system. Yeah. It's a good question to clarify.
Almost look as a reflect a lot about the result.
Yeah, could be. You're being more flexible is better. So, yeah, but this expression of a disorder. So yeah. Okay, some more points. So yeah, this study makes a translation towards more real life kind of tests, and because they were typing in the keyboard and they were getting measures from the typewriting. So, and they also find kind of a reduced habit intrusion during the most automatic syllables. And so we want to go in how to transfer the habits that are in the real world to our lab. So we don't need to train 4 days patients, but they just come with a lifelong established habit, which is much faster and more ecological. So these artificial lab settings have a lot of constraints, like limited rewards, limited time, restrictions, simple context. Is not ecological in a way. So one way is to move and bring these habits from the real world and measure them and see what's going on there. There are some examples that I already have tried to measure them, and they were giving people the keys of their home with different colors, and they had to reverse and change the colors of the keys, and they measure then how many mistakes were doing when they were switching the colors, and they effectively see more errors, like habitual errors when they were switched. So it's really a way to say we can do that in the real world. We can measure it. It's a kind of a very general measure, but these are more clever ways to go outside the lab and get real measures of habits. In this attempt, we did a meta-analysis of what does the brain do, comparing everyday life habits like walking, reading, writing, driving, compared to those experimental habits that I've been telling you about, like learning in the lab. And we explore the brain, and we see different clusters of activity across like 30 studies in this meta-analysis. And we find that the striatum bilaterally is engaged in everyday life habits, and also in experimental, but less so. We see less clusters active in this experimental condition. So it tells us that if we want to study everyday life habits, the striatum is more engaged, like more distributed with all these deep activities compared to experimental ones. Still, there is a adjacent and union on some of these studies. So it gives a bit of hope of saying, okay, the experimental measures can tell us it still activates the striatum, which is our interest. So from this study, we went to do it ourselves with a real life writing task. So we've been writing our lives, less so now with the digital equipment, but handwriting is something everybody knows, and we make them write inside the scanner with 4 different conditions, from more habitual to less habitual, asking them to sign themselves, their signature, writing in Spanish, Polish, or Greek, which is different alphabet to ours. So a transition from habitual to goal-directed. And patients and controls were writing these words while acquiring the different kinematics in a tablet that gave us spatiotemporal variables, which are the typical speed and size, and also some automatic variables, which are based on their stroke. So each stroke can have a very quick and without changes on acceleration times, such as this one. This would be a very automatic movement. But if it's more broken and has variations in the speed and the stroke displacement, then we will have less automatic kinematics. So we have these measures like nib and jerk as part of an automatic index of the writing. There are many more, but these are validated in some writing literature, and we took it for our purpose. And so patients and controls, we had 38 controls, and patients with the left affected hand and patients with the right affected hand. So they were all using the right hand because they were all right-handers, which is a problem for the left Parkinsonian patients. They are not using their affected hand. But we see that they are slower compared to controls, which are in red on most of the conditions, and they have a reduced amplitude, vertical and horizontal. Mostly for the vertical ones, they have this is a smaller vertical amplitude, so the words are smaller because they have a micrographia. So this is all well known in the literature, but we went further and looked into the automatic variables, and what we see is that in the right-handedness patients are the worst, showing the least automatic kinematics across all of the variables. So we were expecting to see more of an effect in the signature compared to Greek, but they were affected in all of the 4 conditions. And then the left PD were similar to controls, but more variability in some cases, but they were not as bad, of course, as the right affected Parkinsonians, and similar in both measures. And when we look into the brain, we see a whole brain analysis. In blue, we see the habitual conditions. So during the signature, there were no group effects. As you can see, it's quite similar. We see an effect of task across different areas and group differences in the cerebellum or insula. Here we see the activity for the goal-directed, mainly in controls, also present in Parkinsonians, and more activity for this habitual condition, which it could be like a compensatory effect of the cerebellum, but we did some connectivity analysis that didn't explain it. We're looking at more deep analysis to see whether there were group differences. We found in the striatum, which was kind of the focus of the study as an area implicated in this task. We see that controls do activate this posterior striatum and quite dorsally, while the patients activate more ventrally and more anterior, which means that they are engaging areas in more effortful condition, in more effortful role. These are areas that are active when you are learning something new, for example. So there's a displacement towards anterior sections of the putamen. Then we also look at the different subgroups. But mainly the message is that there is this transition from posterior to anterior with the overall sample of patients. And we also found like a general, this is non-group specific, like a general correlation with this posterior putamen, left putamen activity with the automatic variables of the writing performance. So it tells us that really this putamen activities is more related when they are more automatized in the writing. So in synthesis, these studies tell us that Parkinson's disease does impact automatic components of behavior, whereas is experimental or everyday, it doesn't matter. It does alter some expressions of it. You have to really look for them. Sometimes, like in the experimental conditions, you have to do tricky things to be sensitive to reveal a difference, as you have seen. In the everyday life paradigm, we see these physiological transitions where normally one would activate posterior areas. They have a more anterior and ventral striatum engagement to do the same task of a very automatized. So this reduced or lost maybe is an exaggerated term, but reduced posterior putamen inputs from dopamine signals could be driven to reduce everyday life activities in writing or other type of habitual tasks like driving. It could also be explained in that way. And this reduction in everyday life habits can be a possible marker of Parkinson's disease. Is possibly more robust than experimental paradigms that are more tricky to perform and have other attentional components and is more messy or noisy. So in this system that it seems to be deficient, depending on how you look at it, what we do with it? Can we train it? Can we compensate with other functions to make them behave up to a normal state? So the question is what to do with this mechanism. So handwriting is very classic problem in Parkinson's disease. So they have been studies where they trained the habit of writing, and they were showing improvement after several days of training. So here you see improvements in several scales. It's too small to see, but these are scales of writing abilities and quality of the writing, which is well, the message is that it was improved. I'm not remembering what I don't see. There are also some other cognitive like fluency scores that were showing a reduction and improvement, and so on. This training was showing recently an improvement of some of the variables in the writing and in the cognitive functioning. So it seems like if you have a lost function, better to train that lost function. It's like going to the gym. If you have a weakening system of your muscles, better to train it than to do something else. That's kind of the take-home message. And with this starting roadmap, we see that in Parkinson's disease, this stimulus response habit seems to be present. Patients still can do their habitual behavior, of course, but with a compensatory brain mechanism that makes them still behave in an automatic and costless manner. So we are still investigating different elements of this behavior with CHEMA and other studies looking at on and off medication, for example, or other ways of habits, not only a motor type of habit, but also non-motor associative forms. And then for another day, we can talk about the impulsivity of how you can regulate or automatize also impulsivity in a way. So I'll leave you here. This work has been mainly done by Lina Guida during her PhD thesis. And this is behind the picture of the new building that some of you know, and you are welcome to come. Thank you for your attention. Questions?
Questions?
Hi, thank you very much. I was just thinking, I don't know if you mentioned that or I lost it. That is there any epidemiological information about risk habits? I mean, habits with a higher or lower risk for development of Parkinson's disease?
There was one paper, not that old, that was looking into job occupations. And they did find a correlation between, I can't remember which job was right now, but there was a link between the type of job you made and the risk of Parkinson. It was not like the message of the paper. It was not the significant one, but you could see a trend. But yeah, it's not super clear.
Yeah, because I don't know, it would be a difficult task to try to map different habits into different connectivity, neuronal groups even, regions and so on. Yeah.
You mean like different type of habits in the dopamine system?
Yeah, maybe, different motor habits, should have different activity representations or connectivity within in the detail. Yeah, I don't know.
The idea is, since you bring this, I think it's an important question. The idea is that if you use a lot your hands during the day, as opposed to, I don't know, somebody that is just thinking or is a less movement-related day, you are at more risk of overusing these neurons. This also is, I open the question to hear us in a non-motor context, such as Alzheimer's disease. It is different. It doesn't work, this type of thinking. The more you use your memory or the more you cognitive reserve or the less risk, right? So it can act different. It can be a different prediction for other diseases, but for Parkinson, we still don't know, of course. But
okay, thank you.
Just to add a bit to the question and the answer, there are several papers that link exercise and physical activity with a reduced risk of developing Parkinson's. There is a lot of interest in this topic, at least clinically, right now. I wonder, because to hear your reflection, because all of these things are also habits, like doing exercise. Typically, it's a habit, and these papers typically refer of moderate exercise, like people that walk a lot, for example. This came to my mind when you were discussing about certain jobs can also involve a lot of walking, for example, or moderate exercises. So how would this like convolute into this type of analysis if intrinsically the habit is beneficial in another pathophysiological way?
Yeah, I mean, it's really limited in a way.
Because writing is also associated with neuroplasticity, and it is known that as a habit is also sort of moderately associated with better prevention in AD, etc. So it's a bit, just to hear your reflection about how this previous habit that is associated with experimental habit may affect the results or not.
I mean, for different types of activities, say sport, writing, are, to me, I don't know these papers that you said, like it can prevent Parkinson disease. To me, is what we do is limited. It's a hypothesis which is very limited to, of course, all the biological, genetic, contextual elements that are in the same pot, right? Of course, otherwise we would all have Parkinson disease because we are all doing every day many different habits. So is limited to just one of the many variables that can lead you to Parkinson disease. We are just looking into one. We don't look at the genetic or the contextual. So, and this about the exercise, I mean, I should check into it because the problem then is that
this can also be actually an inverse correlation. So if the disease is progressing slower for you, you would be able to continue moving. So this association can work. Otherwise, that I find your work actually fascinating because this experimental habit acquisition, I find it very, very interesting to test this.
Well, thanks.
Just I take the ball and then give it back to you. So I think that the relation between exercise and neuroprotection is strong, and is not due to plasticity and stuff. Is more systemic things. So it's such a big effect at the systemic level that all these argument becomes small in terms of it is an habit, it is not a habit. It involves the cardiovascular system, involves so many things. But it's a strong. It's clearly a strong relationship. My question goes more, so you start very strongly as a framework in terms of stimulus-response associations. And then you end up with a task, which is writing, which is not really a stimulus-response association. So I wonder how much of the thinking, because of course, stimulus response is very convenient for some paradigms, for the animal experiments. It's very Pavlovian. So there is a lot of literature on it. But then we don't work all the time as humans in a stimulus response. Of course, you have very strong examples, right, of stimulus response. But how much do you think that the thinking based on stimulus response, which is pretty much open loop, generalizes to a more broad concept of habit, more of in a closed loop type of, which is the way we navigate into the world?
Yeah, first a few things like the stimulus response for writing. When you go to, when do you have to sign? Let's have a thought. They put you the stimulus of a paper, a pen, and you don't think of the response. So in that sense, it is a stimulus response behavior. In our case, it's different because we just ask them to do it. It's not like when you go to a bank and they ask you to sign the document, your signature comes without volition. You just do it automatically, right? But in our case, it's not like that. Of course, we are in a different setup. There is a stimulus that they learn that the experimentalist tells them to do a response, which we believe is automatic because it's signature or writing. And this transition, as you spotted, which is well taken because it's real, we just move from a very solid psychological theory to writing, which is what is writing in a scanner. And the thing is that we think we are in need of novel measures that kind of jump the difficulties of doing a very boring, repetitive tribal trial task that is more controlled. That's the first one, and you get stimulus response, a lot of metrics, bringing behaviors from the outside to the lab. We have to think of which is the best. Of writing was the first one, but we are looking for.
It's not a criticism for the writing. I'm saying
No,
exactly
no.
the opposite. I tend to think that the stimulus response is a nice framework, all nice theory, but at the end doesn't generalize. And as soon as you go to a more natural type of behavior, you enter into the contradictions with the thinking that comes from the framework.
Yeah.
So I think it's much more fruitful to be in a non-stimulus response type of paradigm to actually get to the.
Yeah, I mean, the explanation is we are criticized by using it.
You are criticized.
Yes, yeah. The only paper that we published this meta-analysis. They were like a lot into this critique. Like there is so many sensory motor elements that you are not considering, and while you are writing, there is a feedback of your own body. We just ignore that. But we have to acknowledge that is jumping ahead to get more ecological measures. That's but we gave a more clever response in the paper.
Hi, thank you so much for the presentation. I was wondering, do you think such impairments in the habit system, like the ones you've described now for Parkinson's, could also be in place in Alzheimer's disease? Because often in FDG PET scans, we observe some metabolic changes in the basal ganglia, although they're not the primary sites of pathology. So I was wondering whether you think this could be also the case in Alzheimer's disease, or it could be maybe a secondary dysfunction, maybe because of network impairments, etc.
So as I don't recall, but I should have probably checked before coming here if there is any paper with habit and Alzheimer. But it's an interesting point because I was talking before with Pascual that hippocampus and striatum connectivity is very critical in some of the spatial navigations and the rewarding elements of a task that involves space, memory, and reward and learning. So if there is these two very critical hubs for this behavior, I would assume there would be some form of impairment, but possibly not in such task like the writing, for example. More that has to do with the retrieval of associations, for example, that is very anterior hippocampus related. But I don't recall. I should have checked the literature of Alzheimer.
Yes, thank you.
There is this like almost popular notion that people with moderate impairment like has MCI are very easily disoriented, so they are able to function in their environment. But if you take them to a different place or something disrupts their habits, they got disoriented very quickly. This is a very common notion, I think, in AD. And I wonder, this seems to suggest that habits are very important for these patients that
but they also
rely a lot
get lost in the familiar context, no?
I mean, not so advanced, but people that may be able to still be independent. If you change their context, they will not longer be. So they seem to be relying a lot on their habits for their daily function.
But this is also a question of memory. If you go to a new place, you have to learn new orientations. It's very much depending on memory. And when you go to the common place, this is already in your consolidated memories. I think this could be more related to memory. One question. I mean, Orfeo was thinking about Alzheimer's disease. I was thinking about my kids. I mean, building habits, I think, is something essential. I think it should be something in the school related to that, attention and building habits. You think there could be more scientific approach for education in terms of building good habits, or to kids, for example, or for example, also in patients? I mean, in Alzheimer's disease, there is now a lot of clinical trials for prevention, focusing in changing behavior. I think that could be very interesting, trying to apply a bit more evidence-based methodology in this context of healthy people and or people that are in development.
A very good example in adolescents or children is the use of telephones and screens, and they're trying to put us in the obligation of using them in the schools. With my kids, they're still small, but there is a tendency to start earlier and earlier to use the screens, and that's a toxic habit that we can avoid. So there should be, like, how would you say, high-level decisions to avoid toxic habits from the very beginning. That's one possible. For the disease point, like spatial navigation, I'm thinking currently of a project where you can ask people to walk around a building and get measures, like lab-related measures during a walking real-life habit. Then you could see where and when there is a habit in navigation, and then try to boost it in patients. No, I don't know how, but if we know when are the signals of navigation important, which I guess is turns and cues, no, that tell you where to go, how to potentiate that in the patients could be a behavioral treatment of walking habit. Yeah, just making it up, but it could be relevant. See?
Yes, I want to ask if the improvement that could be with the training, do you think that it's because of the improvement of the connectivity between the regions that are working in healthy people, or because there are other compensatory neural mechanisms that are involved?
In healthy people, you mean?
No, for example, in Parkinson's disease.
So what we see is that it's a compensatory mechanism.
So with other regions, or
Yeah, with a more anterior sections of the striatum.
Yeah.
And cerebellar seems to be there as well. We need to do some connectivity analysis to verify. Regarding the training point in patients, we don't have finalized analysis because all the training task was also done in the scanner at the last day. So if I don't remember wrongly, there was also a kind of a compensatory anterior striatum or ventral. If I understood your question, is whether if we retrain the behavior, we could regain the connectivity.
Yeah.
Yep.
or there is an increase in other regions,
Yeah.
and also if the connectivity between regions that are working in healthy people is also improved.
Is also
Improved.
it's more efficient. There are some studies looking at these training and plasticity mechanisms, and you get just less areas active as long as the more the training, the less areas engaged, and the more energetic, more efficiently energetic connectivity. So you get less areas involved as compared as the beginning. So it is less energy brain wise. Yeah.
Thank you.
Thank you.
Last question. How do you explain in your framework the observation that the Parkinson's disease, for instance, when they are blocked in a habitual task like
freezing, no?
So the external stimulus may facilitate the behavior.
Yeah, that's a good point.
Efficiency.
Well, that after this talk, there are answers before my talk. I mean, this is a very well known, like you activate more externally driven prefrontal areas that are, let's say, sleeping when you get freeze. And that's the hypothesis. They haven't really checked this in the scanner, but you see that that's the hypothesis, that you just recruit areas that are not functioning, that are dose dependent to take the external cues, and then they are, let's say, activate again the walking system. Yeah. So when you put attention to lines, it's just okay. This line activates dorsolateral or frontoparietal network, let's say, and that can get out of the freezing mechanism. That's the explanation, but I don't think like they have done it with brain imaging, for example.
Maybe help you to enter in a goal-directed state.
Yes.
Come back
Yeah.
again, change a bit, and then
Yeah. Well,
everything that you have done is have to
yeah.
kind
Yeah.
of
Yeah.
go back.
The thing is, what if it's freezing?
Goal-directed to
Yeah. To like awaken the goal directed and the attentional areas. Yeah.
Hey, I think it's time. Thank you very much.
Okay.
Great talk.
Thank you.
very much.